I'm TK, and this is TK and Drinks inside the Mead Makers studio, where we explore mead making, get to know mead makers from around the world, and catch the buzz on some of the best meads around. Tonight, we are going to be joined by Nico, the mead scientist, and Dr. Caiti Heil from, I believe it's North Carolina University. I'm super excited to be talking to both of these cats. We're going to be having a yeast discussion, a yeast symposium of something going on, something like that. So looks like Nico has joined me here, so I'm getting him on board. We'll see if we like it too when everything plays nicely. What's going on, Drinking Horn? Appreciate you guys popping in. Maybe? Are you there, Nicholas?
I'm in. Yep.
Mr. Nico, up. Not this time. We're playing that game again, apparently.
Oh, no, really? Jeez. All right.
Of course we are. Well, it looks like Dr. Heil is on board. Can you see me?
I cannot see you from here. Let me,
Uh-oh.
Okay. But I can hear you.
Yeah, so I can hear you as well, and I cannot see you. So these are some of those technical difficulties that we like to deal with. Let's get Dr. Heil on board, see if she can possibly hear us or see us. We may again have to start over at least one more time. Good evening, Dr. Heil.
Hi.
Hello.
It's
And what can you see either of us by chance?
I now can no longer see you, but I can see
Nico.
Nico.
Okay. And what about you, Nico? Can you see anybody?
I can't, but I think everybody can see us, so that's fine.
Okay. Hey, well, you know what? And now it looks like everybody can see everybody except for us seeing each other equally. Hey, you know what? We'll just go with it because
It's all right.
It's all right. So, as I was introducing, getting ready to introduce everybody. So tonight, I usually interview mead makers. We discuss mead products and whatnot and different things. So during a previous discussion with Nico about his meadery Stardust Cellars, I discovered he's a yeast scientist and wanted to expand on this really cool yeast discussion idea. My wife and I had watched a video where people were talking about bread yeast versus baking yeast versus brewing yeast and winemaking yeast. So I wanted to just kind of get this open discussion going and see if we can learn something about yeast from an evolutionary point of view, the history of yeast, and see if we can't just see what we can learn.
Sounds good.
So awesome. So why don't you guys go ahead and introduce yourselves real quick. Start with you, Dr. Heil.
Sure. Yeah. So my name's Caiti Heil. Yeah, you guys can call me Caiti.
Okay, thanks.
To call me doctor. And I'm an assistant professor at North Carolina State University, which is in Raleigh. And I run a yeast lab. I've been working with yeast for about 10 years now. I've been at NC State for about 5, and my specialty is in studying evolution and genetics, and I use Saccharomyces yeast as a model system.
And that's everything that we use is a Saccharomyces for fermenting all of our meads. Some people get crazy with some Brettanomyces, but most of it. And then Nico, if you want to go ahead and reintroduce yourself, never hurts.
Yeah, so I'm Nico. I'm a scientist who study wine and mead microbiology, so the fermentation chemistry, microbiology, and biochemistry of it all and how it comes together. I'm kind of known for the kind of flip side of things where I kind of studied a lot of non-Saccharomyces yeast. I've studied a lot of what kind of the dark side of fermentation, what a lot of commercial meaderies don't really talk about or know about, and kind of what goes on in wild fermentations, and kind of how that differs from kind of genetically modified and engineered yeast strains.
Awesome. So you both have some serious backgrounds in yeast, in the yeast, some serious yeast background. So I'm excited to pick your brains, and let's just start real quick with what is yeast and a little bit about the anatomy and physiology of it, if we could. Starting with you, Caiti.
Sure. Yeah. So yeast. So I'll talk. I guess I can talk mostly about Saccharomyces yeast,
Sure.
but generally, like they're not a very interesting looking creature. They're kind of spherical. They're very small, kind of in the size in between bacteria and human cells. They um, we share a common ancestor with them about 1 billion years ago.
Whoa.
But one of the cool things about them, and the reason why we use them as a genetic model system, is that about 1/3 of the genes that yeast have a similar functional copy in humans. And so a lot of the really important like cellular processes that are happening in our own bodies happen in yeast as well. And so they are used as a system to study all sorts of things. And a bunch of people have won Nobel Prizes for work in yeast, including like cell cycle genes that were really important in understanding how cancer progresses,
Whoa.
and a bunch of other processes as well. And one other interesting fact I'll tell you about Saccharomyces is that recently it's been understood that they Saccharomyces cerevisiae originated in China or Far East Asia, and then and they split from their common ancestor about 5 million years ago, and then they've spread across the world from there.
That's pretty intense. A 5 million year split.
Yeah, so like that's about the same. Yeah, so like they're split from their most close relative is about the same time from our split between human and chimpanzee. But then since then they've evolved and adapted to all these different ecological niches, including fermentations, that make them very interesting to us.
Are they as different from them as we are from chimpanzees? I mean, physiologically?
So yeast look very similar to each other. So if you look under a microscope or on a petri dish, they look indistinguishable from one another for the most part. So the species I work with are actually they're Saccharomyces species, but they're more divergent. So they split 20 million years ago, and if you look at their DNA, it's about as different from humans as chicken.
Wow.
And so, but they look very like identical, but they can act differently in different environments. I guess they have a lot of different abilities to function and adapt to different kinds of environments, and so they while they don't look very different genetically, they're super different. So they're like the sister species of Saccharomyces cerevisiae and its close relative are way more different than at the DNA level than between us and chimps.
Hey everybody, we're back here, TK and Drinks. We got the Inside the Mead Maker Studio Part 2 coming at you with our yeast discussion. For some reason, the live decided it wanted to end prematurely, so I'm hoping we can get everybody back on here, and maybe this time I'll even connect with the video for everybody. So, says Nico is unable to join, except maybe it's a bit. Go live, except go live. Fingers crossed. Maybe everybody connects and we can see each other this time. I
can see both of you.
Oh, I can only see myself, but I'll take it.
And I can see you, Nico, but I can't see Caiti. Okay, so I'm sorry about the technical glitches. If it happens again, I'm gonna I can upload these all and stitch them all together, so we're not gonna really lose much. Maybe just some awkward segues.
I had a question actually. So you were just talking about like unique flavors and that there are some using some different non-Saccharomyces yeast can impart some different flavors than just using Saccharomyces. And I was reading a little bit about what people have been using to make mead, and I was reading a bit about co-fermentation of non-Saccharomyces and Saccharomyces, and I wanted to know whether either of you had ever done that or have thoughts about that or how common that is.
Yeah.
Nico, I'm going to let you field that one.
Sure. Yeah. I can tell you that all of my fermentations are co-fermentations because at Stardust Cellars we do all spontaneous fermentation. So there are no. So it's literally what what's on the honey naturally. So there's going to be a whole bunch of Candida and Pichia. Zygosaccharomyces is a big one. There is some Saccharomyces, but it's actually a very small player in the initial fermentation. But yeah, the way it normally works with co-fermentation is you're going to start off with your heavy hitters first. They're going to take over and eat what's there. Saccharomyces really, is the big showstopper because it's able to, especially with the Crabtree effects, just being able to really quickly eat a lot of, high honey, components in mead. So that really, the osmotolerance of Saccharomyces is really hefty. So it's able to quickly eat and consume, but kind of concurrently, you have a lot of other pathways going on, and a lot of other yeasts that are eating and dominating. Really, it's kind of like a hearty blend of different yeasts and bacteria that grow up until you get to about 7% alcohol, and you start having a pretty major die-off moment between 7 and 10%, where a lot of that, natural non-Saccharomyces yeast, they start to really have trouble keeping up with Saccharomyces. One of the first, original, I guess, evolutionary tactics was figuring out, I have the highest tolerance to alcohol, so it quickly wipes things out, but not before a lot of these non-Saccharomyces are able to produce a lot of really beautiful aromatics, a lot of these funky, yeasty smells that are really beloved in the, natural wine and the funky, the farmhouse beer world. And I'm trying to bring that more into the mead world because I think it has a pretty interesting time and place in mead. But yeah, it works really well. And I always find that Saccharomyces ends up finishing the fermentation. The only thing really left behind at the end, I believe, is Lactobacillus, which happens to be a bacteria with a really low pH tolerance and really high alcohol tolerance. There's only a few strains that survive to the end of it. But it keeps producing a little bit of gluconic acid, which is interesting, and that acid even further prevents spoilage from everything else. And lo and behold, Saccharomyces has an extremely low pH tolerance, so I find that Lactobacillus and Saccharomyces are kind of the strongest bedfellows of the bunch because they always end up hand in hand.
My lab works a lot with sourdough, and Lactobacillus and Saccharomyces are the dominant players in sourdough as well. And the Lactobacillus really, helps create that sour flavor that people enjoy as well.
And that's one of the things I actually discovered first from you, Caiti, was when my wife and I were talking about this whole wanting to start something, that whole yeast discussion. There was I must say an article because it's an understatement. It's a dissertation, I guess, about the bread making and the history of Saccharomyces, and I thought that was a really interesting take on it, and that's why I wanted to talk to you about that. So you in that piece, you guys were discussing that. And you had mentioned previously, and before the video cut out, that the Saccharomyces cerevisiae had originated in China about, roughly, I think I read about 15,000 years ago, and came via the Silk Road. Is that did I get that right?
Yeah, so I think that the timing is kind of having some, there's probably some revisions that are going to happen with that timing piece.
Sure.
I think that it has been hypothesized that humans were the kind of main spreaders of Saccharomyces across the world. But I think that some of our more recent work we look at whole genome, so that's like all the DNA that make up all of the chromosomes of a yeast strain, and we compare them across. People have been isolating and sequencing tons and tons of yeast, all different species, but especially Saccharomyces cerevisiae because it's so important for us. And so we're getting a lot more powerful understanding of the timing, but I think that what it's looking like is actually more that I would like to, I guess I would say yeast maybe domesticated humans instead of the other way around,
I like
where
that.
uh, the yeast were probably in places that humans started using plant materials,
Yeah.
And then the rise of civilization as we know it followed from that.
I got you.
So yeah, so I'm sure that there was trade, but I think a lot of the thought was really biased towards Europe because that has been where most of the microbiological work was done.
Yeah.
Um, so the discovery of fermentation and that it was done by microorganisms and like Carlsberg Brewery were the first people to start actually isolating out single cells of Saccharomyces, and that really kind of allowed people to start fermenting with pure cultures. And So a lot of the work that's been done was kind of focused over in Europe, and there was kind of this thought that Europe and Egypt were and the Mesopotamia were kind of the heartland of the origin of Saccharomyces, but it's now really, really clear from the genetic data that we have that the origin was in Asia first. And there are some pretty ancient like pottery that tells a story about Chinese fermentations happening a long time ago.
That's that's so awesome. I mean, mead is already pretty much touted as the oldest fermented beverage.
Mhm.
And to think that we were fermenting, I mean, not obviously beverages specifically, but fermenting food, and there's instances of other animals, fermenting things or quasi fermenting things. I'm not quite sure what the correct terminology is, but I like to think that um, this whole fermentation thing, like I think you put it best. Yeast domesticated the humans, not necessarily the other way around. Maybe it's a synergistic thing.
Yeah, and I think I think that uh, so one of the stories that has also come out is that uh, our ancestors, our primate ancestors, first developed the ability to metabolize alcohol. So long before the origin of modern humans, um, there was an ability to consume like fermenting fruits,
Yeah,
so fermentation has a start way before us. Um,
even though we hadn't, discovered we were using it not knowing what it was, so to speak.
mhm,
I
mhm.
think
Uh,
Yeah.
it's just a transition to using it externally, really, because if you think about it, we've been fermenting since the dawn of time. It
in,
just, whether it's
in your
fermenting in
stomach,
your
right?
stomach, probiotics, or if it's
Right.
external. So it's kind of our relationship to fermentation goes, I mean, as far back as life goes back, really.
That's a phenomenal way of thinking about it. I really enjoy that. Kind of shift gears here a little bit. So we got a little bit of background in history. We learned that all yeast pretty much physiologically looks the same, little round cells. While they can be vastly different over 20 million years of evolution, they all pretty much get to look the same, which is, I imagine, incredibly frustrating as a scientist when you're trying to identify differences within. Everybody talks about, we say Saccharomyces cerevisiae, but there has to be different subgenres, or I don't know what the correct scientific term would be, different iterations thereof,
Strange.
because
Mhm,
different strains. Thank you. I should have
mhm.
known that. I'm
Oh no.
foolish. Different strains, even farther, because we, as winemakers and bread makers, everybody's got a different, something that they're pitching. So why do we not hear more about those other names, the longer names, the differences, so to speak? Why is it all classified the same? Free for all.
Well, I would say that Saccharomyces cerevisiae has a massive history to it. It's got a cultural reputation to it. It's known as kind of, it's very easy to work with. It's got, a very high tolerance to alcohol, low pH it can work with. It's got a massive tolerance to sulfite. I think that's kind of the missing piece here, is that sulfites really make the world go round. I mean, like 99% of wine has sulfites in it, and mead really, for that matter. A lot of mead has sulfites. And Saccharomyces, get this, it not only has a tolerance to sulfites, it makes its own sulfites. So not only is it out there spitting alcohol, it's also spitting out sulfite. I mean, this thing is insane. So it's literally creating, antimicrobial components as it's churning things out. So you have, a very easy machine to work with. When we talk non-Saccharomyces yeasts, hardly any have any sort of sulfite tolerance at all. Like if I was to add sulfites to my meads, like they would just die off immediately, and all I'd be left with pretty much is Saccharomyces, which is a tactic I recommend for people interested in that. But it's it's kind of a cool tactic. Like, so you have to think like that really whittles down everything that's not Saccharomyces. Zygosaccharomyces does have a small tolerance to sulfite, but not a lot of other components do. Of course, Britannomyces does, because it doesn't care about anything. But it's so that really whittles down 99.9% of all the other choices, and there's amazing choices out there. And if you don't add sulfites, you can actually use a lot of them. So what folks are doing, and this is kind of underground, like it's not really that common yet, but I see more winemakers and mead makers doing it these days, is adding a non-Saccharomyces yeast right off the bat instead of sulfite. And what that does, it starts eating a lot of the potential nutrients. And what you want to do is you create a nutrient desert, is what we call it. You want to knock out everything that could be eaten by something other than Saccharomyces. So the things that Saccharomyces has trouble eating, that's what you inoculate with. And what you inoculate with not only will eat these components that risk spoilage for you, it also produces its own fun mix of unique esters and components, that Saccharomyces might not be able to do. So I see the growth of this industry is happening, but I see it more as a 1-2 punch, co-fermentation, like doing something that will make it easier for your Saccharomyces to be successful. And that's actually a no sulfite strategy, is by using beneficial microbes to do the work to make sure Saccharomyces is happy without having to rely on sulfites and all these preservatives to do the work for you. I think that's more where we're going to see trends happening. But of course, non-Saccharomyces yeast with sulfite tolerance does occur, like Zygosaccharomyces. I think as time progresses, you'll see some genetic modifications happening, people playing ball a little bit more with these non-Saccharomyces yeasts. So it'll happen. But yeah, right now it's kind of limited to this kind of more of like a tech move for wine and mead makers.
Yeah, it's really interesting that you bring up sulfites. You were asking about kind of some of the differences between different strains, and so actually between different strains of Saccharomyces cerevisiae, there's a lot of variation in sulfite tolerance, and the genetic mechanism is characterized, and it's a translocation, which means like 2 chromosomes like broke into pieces and then rejoined themselves. And swapped genetic material, and what it does is create like a higher expression of this gene called SSU1, which creates sulfite resistance. And so just like really when strains have this mutation, it creates high resistance to sulfites, and that's found in all the like wine strains, and it's just naturally occurring. It hasn't been modified by humans. It's just because people use sulfites, this mutation happened and was beneficial to the yeast. And so interestingly, like if we're talking about other Saccharomyces that are not cerevisiae, Saccharomyces uvarum, which used to be called Bionis, also has independently acquired the same type of mutation. It's also used in wine and cider, and again, sulfite is often added to those. And so independently, they've gotten this mutation. I don't know if it's been looked at in Zygosaccharomyces or not. But also, like other wine, like the wine yeast often have like high copper tolerance as well, because vineyards will use copper. They'll spray copper in the vineyards to decrease microbial load, and so wine yeast also have like high copper resistance. And so those are some of the like unique things about wine yeast. So not all Saccharomyces cerevisiae will like be able to do that in a mead.
Yeah, there's definitely a lot of strains and different species of yeast outside of Saccharomyces with more or less an ability to move genes around, and I think that's what makes Saccharomyces so competitive. It's just like HIV, for example, something that's able to move genes around and transpose, like transposons, or just swapping genes really effectively. It's an evolution strategy. So any really hardy yeast, to answer Drinking Horde Meadery's question, they're all eventually going to get sulfite tolerance. So I know it happened with Zygosaccharomyces. It's happening with a lot of Brettanomyces strains. Anything that's really good at picking up genetic drift, they're eventually all going to get sulfite tolerance, because as you mentioned, it's something that does move around. It moves, that gene will move around and bounce around between yeasts. Something Saccharomyces is really famous for is being something with a very high level of not just eukaryote but prokaryote DNA. It's moving a lot, and something very similar to Saccharomyces in that regard is Brettanomyces, and I think that's why you're seeing a lot more Brettanomyces these days, because a lot of them have developed sulfite tolerance.
Yeah, I guess I would describe it as you get what you select for. So if you add any kind of selective pressure, then what will be able to grow in those conditions is things that have resistant to that. So it's not necessarily that it will just spread everywhere, but if you use those, then you select for strains that have that resistance.
Yep.
So we're not necessarily it's not genetic modification, but it's for lack of a better term in my layman person, it's like selective breeding.
Yeah, forced evolution, really.
Forced. What would be the difference? I mean, forced evolution, I guess, in my interpretation of what you're saying, would be we're intentionally subjecting these yeast to certain situations, forcing a change, or basically forcing their hand to change if they want to survive, versus selecting yeasts that are already in the position to do the best job for that and letting them just continue to do that and get better on their own.
Yeah, it's kind of like the difference of whether they're being a human hand and intentionally choosing which species are moving forward to breed. That'd be more what I think with selective breeding. Forced evolution is getting an infinite number of different variable, things with a mass genetic difference all in a pot, having a specific environment or a matrix, and a certain species will outcompete all the others eventually. As infinite generations progress, eventually certain species will outcompete the others by developing some sort of, wild mutation that allows it to outcompete its brethren, just like, a long time ago, Saccharomyces developed an alcohol tolerance, and then that immediately, it built it, and then all of a sudden it's like, oh my God, let's do more of this. And so its genes kind of, were amplified to be more and more ethanol tolerant. And there's no like magical hand doing that. It's just as time progresses, the strong survive, and that's just evolution.
Yeah, right. So so random mutations that are then selected on. And then, so selective breeding per se, I would say like just generally isn't that common in a lot of Saccharomyces, because a lot of what has happened has been this unintentional random mutations that have that the yeast have adapted to these different fermentation environments. And some of those mutations are things like having extra chromosomes or whole extra sets of chromosomes, and a lot of those actually result in the inability to have sexual reproduction. And so that's really actually characteristic of a lot of beer strains, that they cannot ever go through a sexual cycle again. And so breeding is really hard when you can't select parents with desirable traits and mate them to produce offspring that might have traits that you can choose from. And so people have come up with other ways to do this, but it is a lot trickier than it is to work with most of our like crops and agriculturally relevant animal species because of these other weird, like extra hard, like chromosomal differences in some of the strains. I don't know about mead strains if they have, like, a lot of the wine strains are actually um, uh, they experience like natural winemaking processes. Like they're only used in wine for a short period of time, and then they're put back into the vineyard, and during that period they lose access to nutrients, and they often um, uh, go through a sexual cycle, and there may be that may also be facilitated by insects. So insects can actually help facilitate um, breeding in yeast, and
Yeah.
so the one of the differences that's been noted between like wine strains, which I think, from my understanding, commercially people use wine strains in mead production sometimes, and so those strains are much more able to survive in like out in the harsh nature environments, and they can still go through sexual reproduction, whereas like beer strains are just like they are in the lab only.
That's definitely a better answer than I was expecting. um, so, what are some of the biggest differences then between, say, baking strains and brewing strains? Or, I mean, are there? I mean, there's obviously differences, and it seems like through again the forced evolution from people just continuously working with the Saccharomyces and putting it into these situations. Where do we think, I guess, the split is when we really started defining these are brewing strains and these are baking strains, and like kind of what is the difference between those two, if we can?
Yeah, so most of what has made those different strains of Saccharomyces cerevisiae unique to like brewing or wine or baking actually were not intentionally done by us. They for the most part, I think, until more recently when we became aware that we were using Saccharomyces in these fermentations. But they, like, the beer strains have a pretty long history, and a lot of they're characterized by being able to consume these like more complex sugars, so maltose and maltotriose, and those are actually kind of hard for Saccharomyces to eat. So not all Saccharomyces can eat those sugars that are kind of formed together in chains. They like simple sugars that are just like a single molecule. And
Gotcha.
so the ability to kind of break down those is really characteristic of both beer and baking. And then, like, since we were talking about flavor earlier,
Yeah.
there's been strong selection against the 4VG. I forget what the full name of it is. It like has that spicy clove aroma that people generally dislike in most beer and
Yeah.
wine. And so that's been like a known. There's a couple genes that produce that flavor that have what are called non-functional mutations in them that basically knock out those genes, and so you don't get that off flavor. So that has been selected on in yeast strains that are used in especially like particular styles of beer, like ale, lager. You don't want that flavor. You want the clean flavor profile.
Hmm.
And so those are some of the kind of known characteristics. And then I also mentioned, yeah, the sulfite resistance mutations.
Sure. Um, we're you talk you mentioned different desirable and less desirable things. Do we know what the exact mechanism behind different yeast strains providing different desirable or less desirable flavors at different temperatures? Because as you mentioned, like ale yeasts, and we get into lager yeasts and different yeasts that all ferment at different temperatures. Do we know why they have like a temperature preference and then produce the desirables or less desirables outside that range?
I I do know I do know about this, but Nico, I don't know. Do you ever work with different temperatures? Or yeah, do you want to?
Dress says drinking horn.
Yeah. Yeah, I mean, I definitely, I actually work with temperature a lot. So I actually have a facility that's got 3 different temperature and humidity controlled rooms because I have to be really careful about those variables. So my process uses, yeah, a lot of different stages of different temperature control. So when there's a lot of heat, they get really stressed. They have to work overtime. They have to, it just kind of shunts their pathways. It changes the mechanical pathways they have to go through. Like they can't get overheated, so that's going to change their, their biochemical pathways. I know in mead making, the biggest issue I have is fusible alcohol production. So when it gets hot, it stresses the yeast out, and they start producing, these really undesirable flavors. So that's why I use egg-shaped fermenters for everything I do, because it's got a very even center of heat, and it actually causes it to spin kind of like a convex current as it heats and produces its own heat. Because yeast as it creates its own, all of its biochemical reactions, it's creating heat. It starts to cause it to spin. If it gets overheated, it starts to get really upset. So you want to really calm it down and get it kind of, spinning gently. But yeah, it's a big problem in mead making. I'd say more even than wine making, likely because the nutrients are not quite there with mead. Like you have to be a lot more careful. Fruit adds a lot of nutrients, so you're going to really restrict your nutrients, which is going to further stress out your yeast. It's going to start using kind of non-desirable pathways based on those limited nutrients, and if that's coupled with heat, it's really going down some really dark paths, trying to survive, and it can't really use those heat-producing reactions that it would prefer to use. It's instead forced to use other pathways that maybe produce these kind of nasty compounds. That's my perspective, but yeah, I'm sure you got more to say.
Yeah, so I work with temperature a lot in my lab, because there's not that many distinguishing kind of interesting traits between different species of Saccharomyces that are generalizable across the different species, and temperature is really one of the interesting things for me, both from like a fermentation perspective and also from like a natural history perspective, because different species of Saccharomyces have different temperature tolerances. So Cerevisiae is actually, yeah, by far the most thermotolerant. It has evolved to have a much higher thermotolerance than other Saccharomyces species. But that means it has a really hard time. Yeah, I don't know if you have experienced this, Nico, but yeah, in cold temperatures, it really doesn't like to go below 15 degrees and grow well. But other species of Saccharomyces will outcompete Saccharomyces cerevisiae below 15 degrees. And so like lager, which is fermented at cold temperatures,
Right.
It actually is a hybrid between 2 Saccharomyces species. So Saccharomyces cerevisiae and then Saccharomyces eubionis. And they hybridized like without us being aware of it, like about 400 years ago, likely in Germany. And then so now they have like kind of, so Cerevisiae brings like the really good at fermentation, eating those sugars, making ethanol, and Eubionis is cold tolerant, and it basically allows, by their powers combined, like allows the yeast to grow well at cold temperatures, and ferment at those temperatures. And then the I think a lot of people like lager because the clean flavor palette, and when you reduce your temperature, like Nico was saying as well, like when you're really hot, you're getting a lot of stress and off flavors. when you reduce your
Yeah.
temperature, you can get a lot of more nice crisp flavors. So I think there's a lot of interest in people working to kind of engineer and make more yeast that is cold tolerant.
And then you get the crazy Kvike strain that can ferment, above 90 degrees Fahrenheit. So I don't know, was that about 40, 35? My Fahrenheit to Celsius conversion is terrible. But Kvike yeast can ferment incredibly, warm. And that is actually a general question I have, is I don't know what the difference between, is Kvike a Cerevisiae strain, or is it's its own whole separate yeast strain?
Yeah, no, it's Saccharomyces cerevisiae. There's
Okay.
like a lineage that's kind of a distinct lineage. So I don't know a ton about it, but except that I know people are really into it now. But people have
Yeah.
been working to kind of characterize the how it's genetically different from other Saccharomyces, but other Saccharomyces cerevisiae. But yeah, it is a cerevisiae set of like related strains. And
Yeah. It's a lot of fun to play with. There's stories from people from Arizona fermenting with Kvike yeast in their meads outside. I've personally done it and got it up to the external thermometer said it was about 100 degrees, so I can only assume that the internal was significantly warmer than that. And it doesn't produce any off flavors or anything like that. It's such a unique thing by comparison to everything else. So I'm very intrigued by that one.
Yeah, you just have to worry about when you get to a certain level of heat, are you burning off the volatiles in your honey? Do you want
it's
to go
definitely
that hot?
a certain. Oh yeah, it's definitely a certain dance. I like to play a risky game here, Nico. I dance with the
That's
devil in
fair.
the pale moonlight, my friend. So when we're talking about these different yeast strains, when we're baking, I guess we don't really have to consider the temperature so much for the yeast, because by that time, the fermentation is already done or mostly complete by the time the bread or whatever we're baking is going into the oven. Is there a difference in the fermentation rates of the baking strains versus the wine fermentation strains? Because it seems like those baking strains can chew through more complex sugars in a shorter amount of time than these ones that are that we use in the wine and mead making world. It seems like it takes those ones significantly longer to chew through what I would say, or is a simpler sugar, and certainly maybe it's a more sugar type of thing. But I'm curious about that.
Yeah, so I think there are two different main pathways of baking yeast. So you can have sourdough yeast, so yeast Saccharomyces cerevisiae that's isolated out of sourdough, versus like commercial baking yeast that you would get at the store, like Fleischmann's or those little
Like a
packets that
dry
you get.
active yeast of some
Yeah,
sort.
dry active yeast. And those have like very different um, uh, evolutionary histories, and the commercial yeast that you find at the store is much more closely related to some of the beer yeasts, and a lot of them are tetraploid, which means they have so like humans have two copies of every chromosome.
Okay.
In most Saccharomyces cerevisiae are the same. They have two copies of every chromosome. The beer and commercial baking yeast have four copies of every chromosome. And so one of the effects of that seems to be really rapid fermentation.
Okay.
So if you add some dry yeast to your dough, it will rapidly ferment. Sourdough is typically slower. It will take a little while longer to get really going.
Okay.
But yeah, I don't know if anyone has compared like more thoroughly across the different strains besides like those particular known kind of like really industrial yeast.
Seems like the curiosity is certainly outweighing the amount of scientific research that's been done. I don't know if that's because bakers just what I mean by that is like the variation in wine strains. There's you can go to a wine store or a wine website and get 2-3 dozen different strains. At the store you basically get whatever they've got, so to speak, and it doesn't seem like there's a whole lot of variation. Is there a particular reason for that? Do we know? I mean, maybe I'm asking this in the wrong forum, but it just seems like there could be there's a real disparaging difference between the amount of research gone into brewing strains versus baking strains, or baking versus non-baking.
Especially in America, people love their wine and their alcohol a lot more than their bread. I mean,
Sure.
I can tell you there's just a lot more interest in a diversity of yeasts and a lot more flavors and accents. Bread, unfortunately, especially in America, has fallen into this really sad world of being very, very commercialized. So most bread is all about functionality. It's not really about the flavors it presents. It's about how fast can you make a loaf of bread. Because frankly, bread is massively underpriced, especially in the States. So there's kind of been this race to, how do you make the most bread in the shortest amount of time, spending the least amount of money, in a way that wine and beer, luckily, especially with the craft movement, hasn't really been beholden to quite that level. I mean, yeah, there's a push to get the, cheapest price can on the market, but everybody wants that. But there's really just, there's a lot more play. Like, you can have high quality, you can have interesting nuances, you can have 500 different types of wine in a grocery store, but how many people are making bread? There's not really that kind of push for a diversity and diversification versus a push to mass produce, feed the people. It's just a very different industry, I think. I think that's why you're seeing what we're seeing. I mean, I wish the bread industry was more like what it was back when it was an artisanal sort of, industry. But yeah, just right now there's just not that kind of push.
Yeah, I think part of it is just driven by consumer interest, like you said, and so you can create your own sourdough, and I think a lot of what people thought was like that sourdough yeast came from it came from the environment, and that it was like whatever was lying around. But that actually really doesn't seem to be the case from our work. And the sourdough yeast are all more related to other sourdough yeast than they are to the strain that came off a tree in your backyard. And so, but yeah, I guess in some ways sourdough baking yeast strains are kind of the wine strains of compared to like the beer strains in and commercial baking. There's kind of this like wildness to the wine and the sourdough, and then you get like the kind of more commercialized baking yeast and beer. And I guess like you can buy a lot of different beer strains. A lot of them are the same thing.
That's
sold under different names.
Surprising, unfortunately.
Yeah. That's what sequencing, like their DNA, like you can't lie. So that is kind of an interesting thing. There's a smaller subset. But yeah, I think that Nico's point is right. Like the commercialization factor has been really strong.
Well, I've got a good question from. Oh, go ahead, Nico. I apologize.
I was just going to say really quick the word terroir. There's actually a terroir of sourdough, which is awesome. Just like there's a terroir of a lot of yeast strains that are predominant in different parts of the world. Especially France has done a lot of work in creating the terroir of different regions. Champagne has its own cultures and strains that people know really well. So yeah, sourdough is a beautiful thing. It does kind of carry on that terroir tradition, just like the wine world.
Mm-hmm. Yeah, there's a lot of work on French sourdough as well.
There you go. Yeah.
Are the French the ones? I imagine being such a bread heavy culture, and basically France can be almost the center of Western Europe, where you're at. Would they be the ones leading that different type of bread yeast charge, so to speak? You know, Nico, you had mentioned here in America, but like, is there, you know, a greater understanding outside of, you know, our little bubble?
I would hope so.
Yeah, yeah. There's some really prominent scientists working with the different yeasts that are in sourdough, and Delphine Sicard is one that has does a lot of really cool work with like ecology, and they work with a lot of non-Saccharomyces yeast. They're really doing some cool stuff that looks at like how different practices between like farmer bakers versus more like a bakery, how their sourdoughs differ, and the different yeast that may be selected for in those sourdough environments. And they have some cool Kazakhstania yeast that seems to dominate a lot of their sourdoughs. North America, like all our sourdoughs, seem to be mostly dominated by Saccharomyces cerevisiae, but not the commercial baking strains of Saccharomyces cerevisiae. They're unique sourdough strains.
I am now going to be on the hunt for all the sourdough breads everywhere I go. I'm going to get into picking bakers' brains, and let me know everything you know about baking yeast, you guys. This is amazing.
We did try a ferment with the brewery at NC State of some sourdough Saccharomyces cerevisiae we isolated, but it wasn't
good. You tried to make a beer with the sourdough yeast?
Mm-hmm.
Oh yeah, but that was interesting. You had mentioned that at one point that, you know, you can buy all these different types of yeast, and a lot of them end up being the same thing. If you could expand on that just a skosh more, because I'm pretty sure I understand what you're saying, and then I've got a follow-up question for that.
Yeah, so a lot of the beer yeast strains are like proprietary, and they're named stuff like, you know, whatever the company or brewery, W.
Sure.
603 or something, or like whatever they're like a common name. And there's a couple main distributors of yeast. And so some folks like have put on their detective hats. So there was a ton of sequencing work done. So they like took all these strains, they looked at all of the DNA for each of the beer strains, and then compared them to each other. And then like there are some that are called one thing, and another company will call them something else, and the DNA is the same between them, and so not all of them. There are a ton of beer strains, but there are also beer strains that are sold as different things that are the same. And I think like the differences between the different beer strains, I think that, you know, there are genetic differences, but like what really matters is how the brewers, if they are getting traits from those beer strains that are interesting to them.
How the yeast performs is more important than necessarily the genetics behind it, so to speak.
Yeah. I mean, the genetics helps control that, but yeah.
Well, whether if you get the same strain you like, whether you buy it from this brewery named, you know, ABC or this one called 123, you're getting the same thing. It doesn't. Even though they both may have it patented as a different name, it's the same product overall.
Right. Some of them. Yeah.
Got you. Yeah, sure. Not all across the board, but there are instances where there are overlapping genetic occurrences in those types of things.
Yeah, and a lot of them, like, they are all closely related to each other. Yeah.
Other than something from like, you know, it's some of these yeast labs out there, Scott Labs, White Labs, you know, Labland, whoever we're using, other than like their material data sheets or their tech data sheets, is there a place where we can get information for like purveyance and history and genetic analysis of different yeast strains? Or, because as homebrewers and brewers in general, we're pretty much just flying in the dark without, or, with this type of information. Everything's just kind of, well, this is what I heard and that's what I heard.
Yeah, so I mean, there are like scientific papers that talk about this, but they're in kind of technical language. There is this blog that's really great, which I'm looking up right now. It's called Suregork Loves Beer. It's Suregork. And he is a yeast geneticist, and he especially works in beer yeast. So it might not be, quite the same as some of the wine strains and mead strains, but he does some really good kind of breaking down of like new science. Like, I don't think he works on it a lot anymore, updating his blog, but he does have a lot of like the kind of key findings from the DNA sequencing of a lot of commercial beer strains. And I think he does some stuff about the Quebec yeast as well.
Oh, all right, I have to go check that out. I'll be sure to post a link to that when I put this up on YouTube and whatnot. One of the things when I had talked to Nico previously, he was telling me, I was asking him, so, what is your dominant strain? Like, what is the main strain you have there at Stardust Cellars? And he said, bright-eyed and bushy-tailed, I don't know, because he's been waiting for somebody, like a student or somebody, to be able to have the opportunity to come in and do the research for that. Is that something that, like, your lab does things like that, or is that something that would be potential for him? Because I'm saying this because out of a completely greedy thing, because I want to know. Or is that something well beyond, unfortunately, what you guys do?
Yeah, no, we do that all the time. Yeah, no, I run like a genomics lab, so we can grind up the yeast, extract their DNA, and send it for whole genome sequencing. So yeah, you can send us your strains, or we can extract DNA for you. Yeah.
He wants to get a student out there. He wants it to be a cool project for somebody to be able to do and everything like that.
Oh,
he's too humble.
Yeah, no.
Well, he's too humble to say all this stuff. He just kind of wants it to happen spontaneously, and I want it to happen manufactured because I'm impatient.
I've been slowly making the rounds at universities, trying to like get involvement with students, and it's kind of, it's hard, students don't have a lot of time and they're kind of, in the world. But I think Mead has so many research avenues. I just kind of want to start getting more and more students involved and, excited about, starting them. And it seems like low-hanging fruit, like here, just what ferments wild in mead, let's do a research study on it. It seems fun. So I don't want to like take that away before someone like works with me on it, but I'm kind of, I think if I wait another year or two, I'm just going to do it myself.
No, you should do it. Yeah, no, I have not really worked with mead strains at all. And in reading a few papers for this talk, like this conversation, I did get really excited because, in the lab, we grow yeast with 2% sugar. And I was like, I can't believe that they even can handle the amount of sugar in honey. And, I also work with a lot of ecologists that do stuff with bee microbiomes and bee fermentation, and I feel like there's a lot of really interesting overlap there. So I would read your study.
That's awesome. Well, it sounds great. I think we're about 2 hours to the west of you, so I'll have to stop by and say hi sometime.
Yeah, no, that would be awesome.
Yeah.
Yes, making mead science happen. That's what I'm here for.
There you go.
So I guess, I kept you guys for an hour, and I know it's getting late over there on the East Coast. a few more quick questions. What types of studies would you like to see being brought forward on the research level for yeast? I mean, in the near term or in the long term? Caiti?
Um, well, I guess so. My research program right now is in regards to like fermentation specific questions. Like the things that I'm really interested in are um, we have been doing a lot of like genomics work with sourdough. So in understanding just more generally the kind of um, relationships between all Saccharomyces cerevisiae strains. So from nature and beer and wine and sourdough and commercial, and what are the particular genetic signatures that like seem to underlie the unique niche that some of those strains fall into. So that's like a big area of my research right now. And we also do experimental evolution in the lab. So like that's, I guess, kind of like a little bit like what we were talking about earlier, like kind of this like putting yeast in an environment and then letting them evolve for a couple hundred generations, and then we take them and we sequence them and we look at the mutations that have happened. So you can do that with beer or mead or whatever, and you can identify like how is the yeast like mutating during this process. And the other thing that we're working on is looking at, so I mentioned the lager hybrids. We're looking at how like why hybrids can be beneficial in fermentations,
Okay.
because there's a bunch of other, like there's a bunch of wine hybrids. So a bunch of different Saccharomyces species have hybridized and are used in wine fermentation. And I'm interested in like the underlying genetic mechanisms that allow hybrids to be helpful in fermentation.
That's awesome. Like I always thought the more I get into mead making and talking to people who know more about this than me, such as extremely as both of you, I start to think to myself, if making like a wine, like a yeast blend wouldn't necessarily be a bad idea. Would, you know, be beneficial to have, like you were saying, a little bit of bread yeast in there, a little bit of this, a little bit of that, to kind of help each other along, eat the stuff that is less desirable for the ones that you do want, and help it get up there, as opposed to just singling out a single strain and letting it do what it do and being kind of just, you know, everything that doesn't get eaten just drops out to the bottom, so to speak.
Yeah, I mean, I think, yeah, they a lot of times will outcompete each other. But yeah, sorry, Nico, you go ahead.
No, that's fine. Yeah, I think that's really the best strategy to do a co-fermentation. Single strain fermentations are just kind of, I think they've been really played out. I think that's the next big stage in research and really growing the next, you know, wave of wines and meads is, you know, what sort of yeast strains can work together, what sort of yeast and bacteria strains can work together. You know, we're kind of right now at that, you know, back in the day when Chardonnay didn't have lactic acid bacteria, you know, inoculations, it wasn't what it is today. I think a lot of people can agree that there's a huge benefit to co-fermentation in the wine world. You know, buttery Chardonnays have been, I mean, one of the main sellers in the wine world for dozens of years now. So why can mead not be the same? Why is there no, you know, lactic acid bacteria that produces the acetyl in wine? Why is there not like a counterpart in the mead world? I think that's going to be a really fun topic of research.
And hopefully that's something that you can be involved with, and that we can, you know, explore in the world of mead making in the future as a community. I would like to see more, I guess, like you're saying, just more co-brewing, more co-fermenting. You know, people stop the divisiveness between, oh, it has to be this strain or it has to be that strain. This one is great. This one's trash. There seems to be a lot of personal preference out there, and you know, I guess it's understandable about, you know, people are going to have the ones that they like for their certain temperatures and conditions and whatnot. But the part where people get so like in their corner about it, like this is how it has to be and everything. I just like talking to people and experimenting with all these different products, and that's what I'm going to do. I think blending is the new name of TK and drinks game. Blending yeasts, blending post-made meads, blending all that type of stuff. And I'm here for it. So hopefully we can get you guys linked up and get a student out there to start us sellers and get this yeast strain identified and give it a cool name if it's something unique that we've never seen before. I don't know how they name these yeast strains, but I'm here for something unique and wild.
Awesome. That sounds fun.
Very cool. Well, thank you guys very much, both Caiti and Nico, for joining me today. This was everything I was hoping it could be, and I have so many more questions, but I start getting into these questions, I feel that they need more technical explaining, and I don't necessarily have the knowledge to back up a lot of the questions that I have, so I'll have to do a little more digging, a little more research, so when we come up to round 2, I've got a solid background for the rest of my questions.
Oh, this was really fun. Thank you for having me.
Oh, well, thank you again for coming. I know I'd reached out many months ago and been trying to put this all together, but it was the right time, the right place, the right everything. I wouldn't have had it any other way. Thank you guys both very much for being here tonight.
It's been a pleasure.
Yeah. Nice to meet you guys.
Nice to meet you as well. So anybody who's not following Nico over at Stardust Cellars, go check that out. Anybody who wants to know more about cool yeast evolution stuff, go follow the Heil Lab over at North Carolina University in Raleigh. Appreciate you guys very much. Have a fantastic rest of your evening.
You too.
See?
Bye.
Bye. That was awesome. I got to use all my science brain and talk to these guys, nerd out a little bit, and it was awesome. They gave us a ton of great information in there. Next week, I'm going to be speaking with the cats at the AMMA. We have some great conversation there. Definitely a shift in gears from today's conversation, but hopefully nonetheless intriguing and interesting. So thank you all very much for being here tonight. We're going to be wrapping this up. Have a fantastic rest of your evening, and I will see you in the next one. Peace.